So I want you to take a second, right? And just try to imagine the sheer scale of the most abundant biological entities on Earth.
Yeah, it's a tough thing to wrap your head around.
It really is. Like, if you are picturing insects or, even bacteria, you need to think much, much smaller. And you need to multiply that number by an order of magnitude that just completely defies human intuition.
Right.
We're talking about 10 to the 31st power. That is a 1 followed by 31
Which
zeros.
is just an absurd number.
It is. They outnumber all the bacteria on our planet 10 to 1. I mean, they're in the darkest depths of the oceans. They're in the soil of your backyard. They are literally covering the screen you're looking at right now. And kind of crazy they are inside your gut right this second.
Yeah, I mean, 10 to the 31st is a number so staggeringly large it almost loses meaning. But to put that scale into perspective for you, if you took all of these microscopic entities and lined them up end to end, that invisible thread would stretch out into space for over 200 million light years.
Oh, wow. 200 million.
Yeah. And yet, this entire parallel universe of biology is completely invisible to our naked eye.
So today, our mission for this deep dive is to look at this hidden ancient war that is raging all around us and inside us every single second of the day. It's this perpetual arms race between bacteria and their ultimate predator, bacteriophages, or just phages for short.
Exactly.
We're going to unpack the mechanics of what these bizarre entities actually are, how they are quietly shaping the global ecosystem, and why modern medicine is suddenly viewing them as like the skeleton key to surviving the impending antibiotic resistance crisis.
Right. And to understand their potential, we really have to look past the sheer numbers and examine their physical structure.
Which I have to say is genuinely unsettling to look at. If you pull up an electron microscope image of a classic phage, it does not look like a normal biological cell at all.
No, not even close.
It looks mechanical. It looks like a sci-fi microscopic lunar lander. It has this rigid, geometric, 20-sided head, an icosahedron, and these spindly little mechanical-looking legs. And the name itself, bacteriophage, comes from the Greek word phagine, which literally means to devour. So they are bacteria devourers.
Yeah, and the visual of a machine is incredibly apt here because by most strict scientific definitions, a phage isn't even alive.
Wait, really? Not alive?
Not alive. Outside of a host cell, a phage is completely inert. It doesn't metabolize food, it doesn't generate energy, it doesn't move autonomously, and it absolutely cannot reproduce on its own.
So what is it then?
It is simply a highly compressed package of genetic material, so either DNA or RNA tightly wrapped in a protective protein shell, which we call a capsid. It's essentially a dormant piece of molecular machinery that only activates when it physically collides with the right host.
Okay, but if it has no motor of its own, how does it get anywhere? Is it just floating in the microscopic abyss?
Pretty much, yeah. It relies purely on Brownian motion, so the random collisions of molecules to eventually bump into a bacterium.
Yeah.
But and this is key, it can't just bump into any random cell and infect it. The structure of those spindly legs dictates its exact target.
Oh, right, the lunar lander legs.
Exactly. Those legs are called the tail fibers, and their mechanics dictate everything about how phages operate. The tips of those fibers act as a highly complex lock-and-key mechanism.
So they have to fit perfectly.
They do. They're structurally folded to recognize and bind to very specific molecules on the outer surface of a bacterial cell. Things like specific surface proteins or sugar structures.
Which means a specific phage is incredibly picky, right? Like it isn't a broad spectrum killer. It might only recognize and infect one single species of bacteria, or even just one hyperspecific strain within that species.
That extreme specificity is the cornerstone of why they're so medically valuable. And, it answers a question people immediately ask when confronted with the idea of a virus that outnumbers us billions to one.
Which is, can they infect humans?
Right. And the answer is definitively no.
Thank God.
Yeah. Human cells simply do not manufacture the specific molecular locks that a phage's keys are built to open. A phage pouncing against a human cell wall is like, well, it's like someone trying to start a Ferrari with a standard house key. The physical geometry doesn't match, so the machinery never activates.
That is a great analogy. So you have this inert lunar lander drifting until the geometry perfectly aligns, the lock clicks, the phage binds to the surface of the bacteria. But because it's just a protein shell, it can't physically swallow the bacteria, right?
No, it's much smaller than the bacteria.
So it has to somehow get its genetic payload inside and force the bacteria to do the work.
Exactly. And that initiates what we call the lytic cycle. Once the tail fibers bind, the base plate of the phage settles onto the bacterial surface. It triggers a mechanical contraction, almost like a tiny spring uncoiling, and the phage drives a microscopic internal tube straight through the bacterium's tough cell wall and membrane.
Like a needle.
Just like a pneumatic syringe, it injects its highly compressed DNA directly into the host cell's cytoplasm. And the crazy part is, the empty protein shell, the entire lunar lander apparatus, stays outside. It's completely discarded.
Wow. So the invasion is purely genetic.
Purely genetic.
And once that viral DNA is inside, it executes basically a hostile factory takeover.
Yeah.
The phage DNA carries instructions that immediately begin degrading the host bacterium's own DNA.
It's ruthless.
It really is. It commandeers the bacterium's ribosomes and enzymes, stops the cell from performing its normal biological functions, and forces it to read the viral code instead. The bacterium just becomes this mindless assembly line, synthesizing phage parts, stamping out thousands of new geometric heads, tails, and replicating the viral DNA to pack inside them.
And all of this happens at a terrifying speed.
Yeah.
In just 20 to 30 minutes, that single hijacked cell manufactures 1 to 200 brand new, fully assembled copies of the phage.
20 minutes? That is insane. At which point I'm assuming the factory is full and the new phages need to get out.
Right. So the final step of the lytic cycle relies on a specific enzyme produced by the viral code called an endolysin.
Endolysin, okay.
Yeah, and this enzyme literally dissolves the bacterial cell wall from the inside out. The structural integrity fails, the cell bursts open and lyses, and those 200 new phages flood out into the environment, ready to randomly collide with 200 new targets. It is exponential population dynamics at a microscopic scale.
Okay, but wait. Let me push back on this for a second. If these endolytic phages can multiply by 200 every single half hour, the math suggests they should have wiped out every bacterial cell on Earth billions of years ago. Like, it should have been a very short, very one-sided war.
You'd think so, right? But the balance of power is maintained by that exact same lock-and-key specificity we talked about earlier. When a phage population decimates a specific bacterial strain, the evolutionary pressure on those surviving bacteria is immense.
So they adapt.
Exactly. Bacteria mutate rapidly. So eventually, a random mutation will slightly alter the shape of their surface molecules.
The lock changes shape.
And suddenly, the phage's key no longer fits. The bacteria become effectively invisible to that specific predator and begin to multiply again. But, the phages are also mutating, constantly testing slightly altered tail fibers until a new key fits the new lock. It's an endless, oscillating arms race that has been running continuously since the dawn of life on Earth.
That is fascinating, but there's another tactic here, isn't there? Because not all phages operate as immediate assassins. If you're a virus, immediately destroying the only factory that can build you isn't always the best long-term survival strategy.
Right. Some utilize what's called the lysogenic cycle. We call these temperate phages. So instead of immediately commandeering the factory and destroying it, a temperate phage injects its DNA, and that viral code actively splices itself into the bacterium's own main chromosome. It hides there, becoming a sequence we call a prophage.
A prophage, so it functions like a rogue subroutine hidden deep inside a computer's operating system.
That's a perfect way to describe it.
It just sits there, completely dormant. And every single time that bacterium naturally divides and copies its own DNA to create a new generation, it flawlessly copies the phage DNA right along with it. The bacteria does all the replication work for free, entirely unaware that it's passing down a hidden vulnerability.
And this dormant state can persist for countless generations. The prophage only activates when it senses that its host is in physiological distress.
Like what kind of distress?
Well, perhaps the bacterium is exposed to harsh UV radiation or toxic chemicals. When the prophage detects that its host might die, it initiates a self-preservation protocol called induction.
Oh, so it jumps ship.
Exactly. The viral code cleanly cuts itself out of the bacterial chromosome, switches back to the violent lytic cycle, rapidly builds its 200 copies, and destroys the sinking ship to escape.
Man, that is brutally efficient. And because phages are so efficient at hunting and destroying very specific bacteria, early medical researchers realized we could actually weaponize them. Long before we had modern lab equipment, human beings noticed this invisible war and tried to harness it.
We did, yeah.
But the history of how that unfolded is incredibly messy, deeply divided by global politics, and frankly, a bit of a tragic missed opportunity for Western medicine.
It really is. The actual discovery of bacteriophages happened twice, independently, right under the shadow of the First World War. In 1915, a British bacteriologist named Frederick Twort was growing Micrococcus bacteria on agar plates. And he observed these strange, clear, glassy patches forming in the middle of dense bacterial colonies.
So it looked as though an invisible agent was systematically dissolving the bacteria, creating dead zones.
Right. He called them glassy patches, but today we call them plaques.
Hmm.
But he couldn't pursue the research because his funding was pulled and he was drafted into the military.
Wow, terrible timing.
Yeah. But then, 2 years later, in 1917, a French-Canadian microbiologist named Félix Derel observed the exact same clear patches while studying a bacterial dysentery outbreak among French soldiers at the Pasteur Institute.
And he figured it out.
He did. Derel correctly deduced that this invisible, filterable agent was a virus that specifically parasitized bacteria. He actually coined the term bacteriophage, and he immediately recognized its clinical potential.
And Terrell was uniquely audacious, right? Like, he isolated the phages from the stool of soldiers who had recovered from dysentery, reasoning that their bodies contained the viruses that were defeating the bacteria. And before bringing it to the public, he tested his purified phage preparation on himself.
He did, yes.
And then on his family and colleagues, just to prove that injecting humans with billions of viruses was perfectly safe.
Which, thankfully, it was. And by 1919, he successfully used a customized phage cocktail to treat and cure a young boy suffering from a severe, potentially lethal case of dysentery.
So he spent the next decade traveling the globe, championing this new field of phage therapy. And his travels eventually brought him to Tbilisi, Georgia, in 1923, where he collaborated to co-found the Eliava Institute.
Right. And remarkably, despite a century of geopolitical upheaval, the Eliava Institute remains the world's leading center for clinical phage therapy today.
Okay, so if you are listening to this in the US or Western Europe, you're likely wondering, wait, if this was curing severe infections in the 1920s, why did I grow up taking pink bubblegum-flavored amoxicillin instead of a bespoke vial of viruses?
Yeah, it's a huge question. And the divergence really comes down to a fundamental clash in medical philosophy, which was then exacerbated by the realities of the Cold War.
Right.
In the 1940s, the West unlocked the mass production of penicillin and subsequent broad-spectrum antibiotics. And antibiotics function like chemical carpet bombs.
It's wiping everything out.
Exactly.
Yeah.
A doctor doesn't necessarily need to know the exact strain of bacteria causing your ear infection. They administer a standard chemical compound that indiscriminately destroys a massive swath of bacterial life, both good and bad.
It is the ultimate one-size-fits-all solution, mass-produced, highly stable on a shelf, and incredibly profitable to manufacture.
Furthermore, Western medicine was shifting toward the gold standard of randomized controlled trials. You test a standard dose of a standard chemical against a placebo.
Right, the classic RCT.
Yeah, but phage therapy inherently resists this model. Because of that extreme lock and key specificity we talked about, a doctor cannot prescribe a generic phage pill. They have to take a culture of your specific infection, identify the exact strain of bacteria, and then compound a customized cocktail of specific phages that target that exact strain.
And to Western regulatory agencies like the FDA, that bespoke, constantly shifting treatment process just looked unrigorous. You couldn't run a standard trial if the medicine changed for every single patient.
Exactly. So the West largely abandoned clinical phage research. But the Soviet Union, frequently isolated and cut off from Western antibiotic supply chains, relied heavily on Derel's foundation.
They doubled down on it.
They did. The Eliava Institute spent the Cold War refining the art of cultivating and matching phages. The Soviet military heavily utilized phage cocktails for their soldiers. So phage therapy never failed. It simply evolved on the other side of the Iron Curtain under a completely different medical paradigm.
Which is wild to think about. But, okay, up until this point, we have painted phages primarily as microscopic allies, tiny assassins that hunt the pathogens that make us sick. But that sleeper agent lysogenic cycle we explored earlier, that has a terrifying dark side.
It does. It reveals a profound twist in microbiology. Phages do not just kill bacteria. Through a quirk of evolution, they sometimes act as accidental villains by making relatively harmless bacteria incredibly dangerous to humans.
Wait, how does that work? When a temperate phage splices its DNA into a bacterium to hide, it sometimes brings extra genetic luggage, right?
Yes, it carries genes that have absolutely nothing to do with building new phage shells or tail fibers. Instead, these are genes that alter the fundamental behavior of the host bacterium. The scientific term for this is lysogenic conversion.
Okay, but to understand why a virus would carry genes that change bacterial behavior, you really have to look at evolutionary symbiosis. Like, if a prophage is hiding inside a bacterium, its own survival is entirely dependent on the survival of that bacterial host.
Exactly. If the bacteria thrives, the prophage thrives.
So over millions of years, some phages randomly incorporated genes that gave their bacterial hosts an advantage against the host's own predators, like the human immune system.
And the result for humans is catastrophic. Some of the most deadly bacterial toxins known to medicine are not actually found in the native blueprint of the bacteria itself. The instructions for the toxin are encoded strictly in the viral DNA of the dormant prophage.
That is mind-blowing. So the bacteria is infected first.
Right. The bacteria that causes diphtheria, Corynebacterium diphtheria, illustrates this perfectly. A standard strain of that bacteria lacking the prophage is relatively benign. It just causes minor throat irritation. But if that exact same bacterium is infected by a specific temperate phage called the beta-phage, which carries the tox gene, the bacterium begins pumping out the diphtheria toxin. It suddenly becomes a lethal pathogen capable of shutting down human organs.
Wow. And it's not just diphtheria, right? The severe life-threatening Shida toxin associated with major E. coli outbreaks in contaminated ground beef is also encoded by a prophage. And the devastating dehydration of cholera is entirely dependent on a filamentous phage that has integrated its genome into the bacterium.
The implications of that shift our entire understanding of infectious disease. When a patient presents with the severe symptoms of cholera or diphtheria, they are essentially experiencing the secondary effects of a bacterium that is dealing with its own viral infection.
So the bacterium is technically the primary host, and human illness is just the collateral damage of that microscopic integration.
Exactly. The relationship between viruses and bacteria is not just predator and prey. It is a constant, chaotic exchange of genetic information.
And humans haven't just been victims of this crossfire. We have actively scavenged the molecular mechanics of this 10 to the 31st power arms race to build the foundation of modern biotechnology.
Oh, absolutely. The entire field of molecular biology owes its genesis to the study of phages. Take the Hershey-Chase experiment of 1952. Alfred Hershey and Martha Chase were attempting to resolve the most heated debate in biology at the time, which was identifying the actual physical material of heredity.
Because scientists knew chromosomes contained both proteins and DNA, but they didn't know which one actually carried genetic instructions.
Right, and they used the classic T2 phage to solve it, employing a remarkably clever methodology, and believe it or not, a standard kitchen blender.
An actual kitchen blender.
An actual blender, yeah. They realized that since a phage is nothing but a protein shell containing DNA, it was the perfect test subject. They took one batch of T2 phages and grew them in radioactive phosphorus, which integrated strictly into their inner DNA. They took a second batch and grew them in radioactive sulfur, which integrated strictly into their outer protein shells. Then, they let both batches of phages attach to bacteria and inject their payloads.
And the brilliant part was figuring out how to separate the empty shells from the infected bacteria to see what actually went inside.
Exactly. So they threw the microscopic mixture into a kitchen blender. The physical shear force of the spinning blades violently ripped the empty protein lunar landers off the surface of the bacterial cells without destroying the bacteria themselves.
That is so beautifully low-tech.
Isn't it? Then they used a centrifuge to separate the heavy bacteria at the bottom from the lightweight phage shells floating at the top.
And the analysis was definitive. The radioactive protein remained entirely on the outside, in the lightweight fluid. But the radioactive DNA had been injected inside the heavy bacteria and was actively being used to program the next generation of viruses. The blender experiment settled the debate instantly. DNA was the molecule of heredity.
It did. And the biotech breakthroughs didn't stop there. You probably hear the acronym CRISPR constantly in the news, usually framed as human scientists inventing a miraculous new gene editing tool.
Right, CRISPR is huge.
But we didn't invent the mechanics of CRISPR at all. We simply learned how to pilot a defense mechanism that bacteria naturally evolved to fight phages.
So CRISPR is a bacterial immune system.
Fundamentally, yes. It's an adaptive bacterial immune system. When a bacterium miraculously survives a phage attack, it acts like a molecular archivist. It utilizes specialized enzymes to snip out a tiny fragment of the invading phage's DNA. It then inserts that fragment into its own genome, into a sequence called the CRISPR array.
It basically creates a genetic mugshot.
A mugshot, exactly. The bacteria keeps a physical file on the virus.
Yeah.
If that exact species of phage ever attempts to invade again, the bacteria transcribes that stored DNA into a guide RNA.
And that guide RNA acts like a GPS system, right? Latching onto a protein called Cas9, which functions as a pair of molecular scissors.
You've got it. The GPS guides the scissors to the invading phage's DNA, perfectly matching the sequence. And Cas9 chops the viral code to pieces before it can initiate the lytic cycle.
And for figuring that out, Emmanuel Charpentier and Jennifer Doudna were awarded the 2020 Nobel Prize in Chemistry, not for inventing the system, but for recognizing that we could program it. By synthesizing our own custom guide RNA, we can direct the Cas9 scissors to navigate human DNA and make precise cuts at any specific genetic sequence we choose.
It's revolutionary. And actually, the 2018 Nobel Prize in Chemistry was also born from this microscopic war. That one was awarded for a technique called phage display.
Phage display, how does that one work?
Phage display is an elegant manipulation of the phage's physical structure. Scientists learn how to physically splice a specific foreign gene directly into the viral code that builds the phage's outer protein shell. Because of this linkage between genotype and phenotype, when the phage is assembled, it literally displays that foreign protein on its exterior.
Oh, wow. So researchers can create vast libraries of billions of different phages, each displaying a slightly different protein variant on its surface.
Yes, they can wash this massive library over a specific target, like a human cancer cell or an inflammatory marker. Most of the phages just wash away, but the ones that perfectly bind to the target remain.
And then they isolate those specific phages, read their internal DNA to see exactly how that successful protein was built, and use that blueprint to manufacture highly targeted medications.
Exactly. This is the exact underlying technology for some of the most effective therapeutic antibody drugs currently treating autoimmune diseases and cancers today.
It is wild how many Nobels just come from scavenging the molecular debris of this microscopic war. But, our primary interest in phages is rapidly returning to their original function, killing bacteria, because the era of the antibiotic carpet bomb is nearing its end.
It really is. Think about the last time you took an antibiotic. We have spent 80 years applying massive evolutionary pressure to bacteria by drenching our bodies, our livestock, and our environments in these chemicals.
And the bacteria have adapted.
They have. We are currently facing the rise of superbugs, strains of MRSA, or highly drug-resistant Pseudomonas that actively neutralize or pump out our strongest antibiotics. The pharmaceutical pipeline for discovering new classes of antibiotics is effectively stalled, and global health organizations are warning of a rapidly approaching post-antibiotic era where a simple scrape could once again be a lethal threat.
Which is terrifying. But this impending crisis is forcing Western medicine to finally look back to Felix Durrell and the century of work preserved at the Iliava Institute, right?
Yes, finally. We are witnessing a massive resurgence in clinical phage therapy trials in the United States and Europe. We are already seeing numerous well-publicized compassionate use cases where patients dying from completely untreatable multi-drug-resistant bacterial infections were saved as a last resort by customized cocktails of purified phages.
Because phages replicate and mutate alongside the bacteria, they offer a dynamic, living weapon against resistance that a static chemical pill simply cannot match. If the bacteria mutate to avoid the phage naturally mutates right back to catch it.
Exactly. And the applications extend far beyond hospital walls. Because phages cannot bind to human or plant cells, they are incredibly safe for human consumption and the environment.
I read that the commercial food industry is currently utilizing FDA-approved phage sprays on fresh produce and ready-to-eat deli meats.
They are. These sprays specifically hunt down and lyse pathogens like Listeria and dangerous strains of E. coli without requiring harsh chemical preservatives or altering the taste of the food at all.
And they are also being aggressively researched as alternatives to broad-spectrum antimicrobials in agriculture and livestock management, which is currently one of the massive drivers of environmental antibiotic resistance.
It's a hugely promising field.
So, we've spent this deep dive exploring the terrifying efficiency of their mechanical anatomy, the historically divided legacy of their discovery, the counterintuitive reality of lysogenic conversion, and the biotech revolution they provided us. Phages are the invisible architecture of the microscopic world, and they may just be the cavalry we desperately need.
I couldn't agree more.
Thank you so much for joining us on this deep dive. But before you go, I want to leave you with one final thought to explore on your own. You have likely heard extensive discussions about the human microbiome, the trillions of bacteria living in your gut that profoundly affect your digestion, synthesize your vitamins, regulate your immune system, and, according to recent neurological science, heavily influence your moods and behavior.
Right, the microbiome is everywhere in the news.
But consider the scale we discussed today. If there are 10 times as many phages in your gut constantly hunting, regulating, and transferring behavioral genes between those bacteria, are your physical and mental states actually being secretly puppeteered by the trillions of viruses in your phageome that humanity has barely even begun to map. Next time you trust your gut, remember you might actually be trusting a phage.